session. And just so you know, you're in the right place. This session is entitled Monarch Citizen Science for All Ages. And our speaker today is Mary Beth Cary. And she began learning about environmental education in 1995 after being charged with developing an outdoor classroom at Worth County Primary School, thus beginning a 20-year immersion in content knowledge, lesson development, and best practices for outdoor education. She was in the first AT cohort and qualified for National Board certification in 2005. When she retired in 2015, she began fulfilling her plan to volunteer with Monarchs across Georgia, which is one of environmental education's wonderful initiatives that you may already be familiar with. Just some basic housekeeping. It looks like everybody's already muted. That'll help with any background noise. Trying to moderate another session, so that was fun. But if you have any questions during the presentation, feel free to put them in the chat box, and I will probably wait towards the end to go through those with Mary Beth. So take it away.
Okay, good morning all. Karen, I know you already know all this stuff. I don't even know why you're doing this, but I will refresh you. We have somebody else coming in. Good morning, Katie.
Good morning.
I'm going to tell you about how to test for monarch parasites and how to tag butterflies. And I actually caught a monarch yesterday, and so hopefully he's still alive. I put him in the refrigerator, and he was kicking around a little bit ago, so hopefully he will have survived the episode. He'll have something to tell his peers. First of all, with Project Monarch Health, the OE parasite, I'm not even going to try to say the name of it. Linda's going to put up the monarchhealth.org website. And if you see a monarch that it closes, that means comes out and it's all crumpled up and stuff, then it is sick. But lots of monarchs are infected, but you can't really tell that they are. So, and they can have spores that spread, and the mama has it on her abdomen, gets it on the leaf, then the caterpillar eats that leaf, and so on and so forth. And lots of them are not making it to Mexico because they're infected and not strong enough. But then they're studying how many are getting there and getting back and all that stuff. So the things that you get when you ask them for the sampling kit, that's at monarchhealth@gmail.com. The supplies they send you are blank index cards, clear tape stickers, an envelope to mail the samples back, the instructions, and a data sheet. And the data sheet is real important that you fill this out correctly. Then you also need some gloves, a 20% bleach solution, which I'm keeping way over here because I don't want it to get confused with my water that I might be drinking. And to make the 20% bleach solution, it's a teaspoon of Clorox or bleach to a cup of water, because I wasn't making a ton of it. So I just wanted to get it down to a manageable size. Okay, so to first fill out your OE sampling data sheet, you have at the top, you have your lead observer, which would be myself, team members if there are any, the email, I haven't quite filled that out yet, the collection address, and this is real important that they know exactly where you were collecting. And for if you change sites, I want you to do a separate sheet. And you can put the coordinates in too. They ask if you have milkweed around, what types. And then down here, they have the ID number. Now I use a mixture of ID numbers because I'm also tagging, and I use that ID number on some of them and then just a regular 1, 2, 3 on others. You date collected from the wild. If you collect them as caterpillars, you put down that date. If you if you catch it wild, you would put down the date you caught it. So like I put down 9/16 for this monarch. Stage when collected from wild, that's egg, young larva, old larva, pupa, or adult. So you put where it is, and then the date sampled for OE and the sex, whether it's male or female, and we'll talk about that in a minute. And then any notes, like on some of mine, I could tell they were sick. I sampled them because just to see, so they could see how sick they were. But I had to euthanize some, and that's what you're supposed to do, is put them in the freezer if they're not going to make it, and not keep them as pets. Okay, so to sample for OE, we're going to put the gloves on. If I can get I should have put my gloves on ahead.
And Mary Beth, is this sampling just something anybody can do that has monarchs around?
Oh yes, this is, and when I taught, I had my my little ones, my first graders, doing this. You know, they had to have a little help, but any age group can do this. So, you know, you might want to help them some, but the reason why I've said citizen science for all ages, although now it's being called community science, because kids really like being scientists and sending data off. And, you know, this is going to be used by the scientists at the University of Georgia, you know, and they think they're really, really something. And it also, you can emphasize to them how important collecting the data is, that they need to be very concise in that, and that you don't remember it later. You have to write it down. Okay, I'm gonna get him. I caught him yesterday, put him in a glassine envelope and in the refrigerator, and he's still kicking. You can see his little feet just a-going. Usually I just use one glove, but today, because I don't want them to fly off and all that stuff, I'm gonna use two. Okay, to test them, when you hold them like this, they stick their abdomen out. You want to, let me get a good grip. You don't want to get their legs, but you want to kind of wrap this around their abdomen, remove it, and you have little cells on it. Now you can look under a microscope and see they look like little bitty footballs. That's how they're different than the regular cells. Then you're going to put it on your index card, and they want you to put it on horizontally. It used to be, it didn't matter. And I urged, so see, that's how I have been doing it, and found out when I watched the video that that's not the way they want you to do it anymore. So I would strongly suggest, even if you've been doing this for a long time, that you go back and watch the videos, because some things have changed. Okay, so I'm going to use my tag number on this, and it's 963. So I'm going to write 963 underneath it. You know, you can number them however you want. And now we need to know what sex it is. Well, to find that out, you get hold of the feet and you let them open up. And this is a male because he has 2 dots there, the sense dots. A female would not have that. He's still getting one of his wings kind of mucked up. So we're going to write that down as a male, and we'll record that on our sampling sheet. Okay, the next part then is to tag the butterfly, and the tags are little bitty stickers, and it's amazing that they can stay on. So I'll tell you what I do is use a toothpick to pop them up, because especially when you've got gloves on, you know, everything sticks to them. So I'm going to pop up this tag, and there I have it on the toothpick. And you're going to put it on the cell that looks like a little mitten. See if I can hold it up right. Okay, you see the cell right near the end of my thumb. It's the discal cell. It looks like a little mitten. And so I'm going to put it on there and kind of hold it down. Now, this does not hurt the monarch. Monarchs are really tough. I mean, they fly all the way to Mexico and back. By putting it on this cell, it kind of keeps their equilibrium. So they're not, you know, don't have weight on one side and not the other and flying wonky. And then the other thing you need to do, and even if you're not tagging them, I do this, is mark the monarch so that you know you've already sampled it. Because if you're out running around catching monarchs in your garden, then you don't know whether you've sampled them or not, you know, because they kind of all look the same. And so you take a thin Sharpie, excuse me, and you want to put just a little line on that cell. Okay, I just, right near the tag, I just put a little line right there. And that way, if you catch it again, or if it loses its tag, then you know that you've already sampled it. Okay, so I'm ready to let him go, but I'm not going to do that because I'll have to get up and walk away. So we'll put him back in his little envelope for a little while before we send him off to Mexico. Now, the male, to sex the monarch, the males, as I said, have that not cooperating. Of course, I wouldn't want to cooperate if I've been stuck in the refrigerator all night either. Okay. Then you would take your bleach water, clean your surface, also clean your pen, because you have been, you know, you have the stuff on your, if it has parasites, you get it on your pen because you're touching it. So you want to do that. I'm sorry, I'm bouncing around as things come to my attention here. All right, to a female, now this is a deceased one, so it's a little crunchy. Oops, that says male on the package, but it's a female. Because, let's see if I can get a hold of it. It has those, oops, it's very crunchy. Can you see it now? See, that's a male. So I have a pile of them here. These are monarchs that have been used in the, I'm not sure if they came from the Alzheimer lab or they have work with them at Emory also, and that's where Susan gets her samples when we do trainings. Okay, this one, I'm holding it upside down because that's the only way I can get it and show this to you. The females have thicker veins, of course. They're usually a little bigger, and they do not have that scent gland dot on them. Okay. So this is taking a lot less time than I thought it would. So we have time for lots and lots of questions. Yes, Kitty, I do change gloves between each one. That is a recommended practice. Karen marking them. Okay. And the tags come from monarchwatch.com. I think it's .com, isn't it? No. Yes. monarchwatch.org, excuse me. And then the sheet that you get with the Monarch Watch with the stickers, they'll have an extra sheet that explains to you where to put the sticker. And then, of course, the monarchs will fly away to Mexico. And when some are deceased and there are piles of them on the ground, especially if they've had a cold spell down there, and they pay the ejidos, the locals, to collect monarchs, and they pay them like 5 pesos each for ones that are tagged. So it's a really big deal if your monarch is found down there. You know, it's like, oh, and this is how they track where they had come from. Because, and with the Monarch Watch, you can fill out a spreadsheet to send your data in electronically. With the OE, you have to send your sheet and your samples, of course, because you can't do the samples of the cells electronically.
Mary Beth, we got a question from Mike. He's wondering if OE sampling is done during both spring and fall or just fall. Does it matter what time of year you do it?
You know, I think no, it can be done year round, and there are year round populations, especially in Florida, and they're just starting a new study looking for roosting sites along the coast of Georgia. But I have sampled in the spring and summer. Where I live in South Georgia, you know, for some years you don't have any monarchs, and then some you do. So yes, you can sample throughout. They also are sampling soldier and queen butterflies, and explains that on the website because they're trying to see if this parasite, how widespread it is among other types of butterflies.
All right. And without testing them, is there any way to know just from observing them, are there any visible symptoms that a monarch might display that has OE?
Okay, they may have like a crumpled wing. Sometimes they look okay, but they can't fly. It's pretty obvious if they're infected a lot, but like this one that I just tested, no, we can't tell until we look at this sample under a microscope. And you can do this with your students too. You can look at the sample, and there are examples on the website of what you're looking for. I've tried a magnifying glass and it doesn't really work that well. So I think you want something a little more powerful to do it. And you throw the toothpick away and get a fresh one the next time.
What about caterpillars? I know when I visited Kitty Spivey down at Pebble Hill Plantation recently, we saw a caterpillar that had died. Just kind of, you know, limp on the milkweed. It turned dark. We were kind of curious what might have caused that. I mean, there could be a million things, but
right. There are a lot of
OE and caterpillars as well.
Yes, and then also usually when they're black and hanging like that, it can also be that a wasp has come along and sucked the juices out of it. There are critters that will parasitize them. Taconid flies will lay their egg in the caterpillar, and then if you have them in captivity, you can see the larva, I guess it is, comes out and it makes like a little thread and goes down. And then the chrysalis turns dark. And that's another way. If your chrysalis is turning dark, that could be a variety of reasons, OE and parasitization. It's been parasitized. So you never know. It's fair. Some of them with bad OE, it kind of makes them explode. Yeah. And like have diarrhea or something. It's kind of yucky. And I feel sorry for them too. On the milkweed, we are strongly encouraging people not to plant the Quercevica, the tropical milkweed that you can get in the big box stores. It's very highly available, but unfortunately, it can encourage the monarchs to stay longer because it does not die back like the other varieties do, the incarnata or the tuberosa. And if you have that Quercevica, they strongly suggest that when the winter comes, when it starts freezing, that you cut it down so that the monarchs would not have anything to eat, the caterpillars. Years ago, before we knew all this stuff, we were so excited because we had a Quercevica plant outside my classroom door and had a caterpillar in December. Well, now I know that's not a good thing. You know, if it made it through the weather to become a butterfly, what was it going to eat in the winter time? So that can be a big problem. They also would prefer you not to plant Syriaca, that's the common milkweed, because up in the northwest corner of the state, I believe it is cross-pollinating with an endangered milkweed. So they don't want that to get loose. And the Syriaca spreads a lot. And it has nice, fat, juicy leaves. So that's sad that happens. If you're going to plant milkweeds to encourage your caterpillars and all, you need to plant a multitude of them, because the scientists will say, well, the mama just lays one egg per plant. Well, I have not found that to be true at all. I guess it's because there aren't that many milkweeds around for them to lay on, but they'll munch up. One caterpillar will munch up a whole big plant in no time because they are eating machines.
And if you see a caterpillar that's on a milkweed that is completely void of leaves, if you don't move it to another plant, does it just die? Well, how does that work?
Lots of times they will move themselves because they go a long way, especially when it's time to make their chrysalis. You'll find them, 10 feet away from where they were. It's amazing. But I'll move them a lot of times, or sometimes they'll continue to eat on the stem. But I feel responsible. I feel like I've invited them to dinner, so I have to feed them. So okay, here, move over here and you can have some more.
And these are all my questions, and hopefully others have them too, so I'm not just boring everybody. But to get the OE kit, does that cost anything?
No, it's free. You just do the email
monarchhealth@gmail.com.
Correct. And tell them you want a monarch tagging kit, and they'll send you the index cards, the stickers, the direction sheet, data sheet, and you can also get that online. And that way, if you, like, mine is kind of messy because I'm, juggling monarchs and trying to write and all that stuff. So I redo it before I send, so it looks nice and neat, so they won't be saying bad things about me or not being able to read my numbers. But all that stuff except for the bleach water and the gloves and the Sharpie. Those
And the
things
toothpicks.
those are not included. You have to.
Right.
Oh yeah, toothpicks, but that's a different thing. That's with Monarch Watch.
Oh, yeah.
Talking about
See, I'm getting my wires crossed.
Monarch parasites. So yeah,
Right.
it's
We
confusing.
do have one other question. Are monarchs like hummingbirds in that they somehow pass on the locations of milkweed from year to year? So like hummingbirds, if you have a feeder out, it's been documented that they will come back to the same feeder. Do you know if that's the case with monarch butterflies as well? Even though it's not the same individual, it's obviously part of a series of generations.
They're just wherever the wind blows them or wherever they're floating around. And the book gotta go to Mexico. Well, the females are gotta find a milkweed to lay my eggs on, so they don't have any homing abilities, but they do have the ability to fly from Canada all the way to Mexico in the fall to overwinter in the Oyamel forests in Mexico.
But they're just not particular on coming back to the same garden necessarily. It's just planted and they will come. Some of them will find it.
Right. That's the way it always is, plant it and they will come in any garden. But they don't have any homing device or pass the word on to their friends.
All right. Any other questions? We've only got 3 minutes left. Speak now or forever hold your peace.
And on the, you can save the chat if you go down, if you want to save the
links and everything.
Yeah, let me figure out.
Yeah, you just go to the bottom of the chat box, click on the 3 little dots there. It'll say more, and then at the very top of that pop-up menu will be save chat. And it's basically like a text file that it'll save as.
Yeah, and that way you can have the websites. Should have told you that ahead of time, but oh well.
We got them.
For the next event, you can do that.
All right. And also, you guys, if you want to stay in touch or possibly even volunteer for Monarchs across Georgia, I'm putting in their URL now. Of course, all of this you can certainly just Google. And even though, they have monarchs are kind of the poster child for pollinators, but certainly when you help monarchs, you're helping all kinds of pollinators that are so important to our ecosystems.
And on the Monarchs Across Georgia webpage, there is a plethora of information and links to all kinds of stuff to fill you in. There's a milkweed field guide. There's a list of nurseries.
Mike's asking real quickly if you can mention in something about symbolic migration.
Journey North could no longer fund that. So Monarchs Across Georgia has taken that over. And we did it virtually this year because, everything has to be done virtually. And classroom teachers buy a ticket that covers Estella in Mexico, our contact who reports on Journey North. And she goes from school to school with books for them and teaches a lesson in ecology, and each year it's a different subject. And she takes those class butterflies that they have made to the children at the schools in Mexico, and they get to keep them. And these children have nothing, so they're so excited to be able to have a butterfly from America because there's a little sheet and it tells exactly where it's from. And then there are individual butterflies that the students decorate, and those are shuffled around and sent back out to the schools. So classrooms get butterflies from all across the country and Canada, and they can track those.
Very cool. I hate to cut you short, but we're out of time.
Okay.
We're going to let folks move on to their next session. We've only got just one more after this. So thank you all for joining today. And this session will be recorded or is being recorded. So you can always go back and watch again. And like Mary Beth mentioned, you can save the chat box for all those resources as well. So thank you for joining.
Thank you much. And thank you, Linda.